Alright, so now what I'm going to talk about is protein purification. At this point in time, we've talked about individual amino acids, we've talked about a little bit, we've talked about polypeptides and the overall structure of a protein. But now what we need to get into is kind of an experimental part of the class where we talk about how to actually isolate a protein in order to characterize it, which is one of the key parts of any sort of biochemical or protein biochemical research. So if you would please pay attention to this and we'll get through that discussion. So the important parts of knowing how to purify a protein are really, well, before we get started on that, you need to know some definitions. First of all, any protein is a polypeptide that is 50 amino acids or larger. Anything smaller than that is simply called a polypeptide. So if you have a polymer that is 45 amino acids, that would be known as a polypeptide. Now when it comes to purifying proteins, what you're doing is you're trying to take a complex mixture and basically distill it down. Or not distill it down, but separate it from the vast majority of the components. And so what that means is you are going to have to take a complex mixture of all these different proteins, and I'm just going to make it kind of simple. You're going to take this mixture right here that has, well, basically 3 different colored globs, blue, red, and green. And what you want to do is you want to extract only the red ones. So how are you going to do that? And this is just a visual example. What you're going to do is you're going to go through a series of steps. The first step that you need to do is you need to solubilize your sample. So you need to put it in a buffer in which your proteins are, in theory, going to be functional, but more than anything else, that your proteins are going to be accessible. They're going to not be in an insoluble fraction. Okay, so this is done through a number of different processes. One, you could use a blender and just blend up the tissue, just basically blow it up more or less so that it's a homogeneous solution. You could also use an instrument known as a French press, and that is very similar to a French press that you would use whenever you're making French press coffee. You're putting a high amount of pressure onto your sample, and you're again just more or less like rupturing those cells open. Okay, the next thing is centrifugation. So with centrifugation, what you're going to do is you're going to take a, let's just draw a microcentrifuge tube here. You're going to take a mixture that has all these different proteins in this homogeneous mixture, and what you're going to do is you're going to produce the following. Well, here is after centrifugation, you have a somewhat transparent solution, and then you've got this pellet, this bunch of junk at the bottom. So you have a soluble fraction and an insoluble fraction. Now, whenever you're doing this, what you always want to be able to do is you want to have a means to detect a protein. So the first way to detect protein is, well, using the presence of our aromatic amino acids. Aromatic amino acids will absorb light at 280 nanometers. And so what you can do is measure or take a small sample of this, an aliquot of this, and measure, do you see any absorbance at 280 nanometers? Do the same for your soluble fraction after the centrifugation. Now, in the event that you see A280 here, or you see an absorbance at 280 here, but then you don't in here, well then you probably don't want to use that soluble fraction. That means all your protein went to the insoluble fraction. Chances are slim that that's going to happen. In reality, you're probably going to see A280 in both, or you're going to see absorbance of light at 280 nanometers in both of those samples. Okay, so another thing that you would want to do is you want to be able to detect your protein. Now, a good way of doing that is by knowing what your protein does. Now, I'd like to think of a a simple example of, and not even, just a simple conceptual example. Let's say that you have a protein that takes circles and makes squares, okay? Now let's say that these circles, or let's say that the squares absorb light at 340 nanometers, NM. Now, if those squares absorb light at 340 nanometers, when you see absorbance at 340 nanometers, you know that there are squares present there. So if you are looking for your protein of interest that absorbs, that produces a product that absorbs light at 340 nanometers, well, you want to see it here, and you want to see it in that soluble fraction from here. Okay, so that is kind of the foundation that you'll use to go ahead and begin processing your samples. So if you have a fraction that does not absorb light at 340 nanometers, well then that's your indication that your protein is not present, because you're not seeing the production of any of these squares. These squares are the products that generate or that absorb light at 340 nanometers. So if you have that, that means to detect protein and to detect your protein specifically. This is a very specific this is a specific element, then you're going to be in good shape, okay? So when you have all that, then you can move on to, well, using some chromatography techniques. Now, after you've collected your soluble fraction from the centrifugation step, you can begin doing some things with that. So you have a soluble fraction. And with this, you can go in a couple of different directions. One, you can do a process known as desalting. This is also known as protein precipitation. And you can use a salt like ammonium sulfate. Ammonium sulfate is a highly soluble salt, and so what that means is whenever you put it into a solution, it goes very easily into that solution. Whenever you centrifuge a sample, so let's, again, here's our microcentrifuge tube. Both of these have ammonium sulfate. This one right here has been centrifuged. You have this pellet at the bottom. Okay, that pellet at the bottom has a ton of this ammonium sulfate. It has precipitated out. And with that, it's going to go ahead and grab onto some proteins and cause those proteins to precipitate. So ammonium sulfate precipitation is a very common technique. The great thing about this is, again, thinking about our product, absorbs light at 340 nanometers. Well, in this sample right here, our soluble fraction, we have not done anything to this. We just centrifuged our crude extract. We see, yes, indeed, we've got production of our product, absorbance at 340 nanometers. That's a great indication. Now, in our second sample, well, now we've got basically a separation. We've got an insoluble fraction and a soluble fraction. Now, if you see absorbance or if you measure the absorbance at 340 nanometers, and in the soluble fraction, you don't have anything, what does that mean? Well, the great thing is, that means that your protein has been basically picked up and is in this dense salt crystal. So the great thing about that is you know where your protein is. You know that it's not in your soluble fraction. You know that it's densely packed with all of these different ammonium and sulfate molecules. So you know where your protein is. And so this is a fine thing to do. Now, in the event that the opposite is true, you see all your activity in the soluble fraction, that's fine as well, because have you gotten a good number of proteins to precipitate with that ammonium sulfate? Absolutely. Okay, so it's commonplace that after you do an ammonium sulfate precipitation, you go on and you start doing some column chromatography. So let's say that you do column chromatography, and there are basically three different techniques. There's gel filtration, there is ion exchange chromatography, IEX, and then there is affinity chromatography. Okay, so in gel filtration chromatography, this is going to separate by molecular weight. And we draw something known as a chromatogram. This chromatogram measures on the y-axis basically absorbance at 280 nanometers. Is there protein there? On the x-axis, you look at fraction numbers. So 1, 2, 3, and so on. So what you will see with a chromatogram, and what is commonplace to see in a chromatogram, is something that looks like this. Extend that line. What this shows is that in the first 3 fractions, you have a lot of protein there. Now, what do you know about those proteins? Those proteins, you know that they are high molecular weight. And the way that you know that are by internal controls, where you would have something that you know the size of it. And let's say that it comes out in fraction number 3. This maybe absorbs at a different wavelength, or you have some means of detecting that molecule. And let's say that that molecule comes out in fraction number 3. It is 175 kilodaltons. You can compare the fractions in 1 and 2 and say, well, I know that they're larger than 175 kilodaltons because I know the size of my internal control. Then you'd also want to use a second internal control. Coming out in this fraction, let's say it's 45 kilodaltons. So whenever you're confronted with something like this, with these 2 bookend molecules or proteins, you know that anything that comes off of the column after the 45 kilodalton or before the 175 kilodalton fraction, you know approximately the sizes they are. Or at least you can say it's larger than 175 or smaller than 45. What that means, and the benefit of that, is if you are trying to purify a protein that's, say, 120 kilodaltons, well, chances are, when you see these 2 peaks right here that I've indicated with green, well, one of those 2 peaks could be your protein, because you know that your protein is smaller than 175 and larger than 45. And you have these 2 bookend proteins, or 2 bookend internal controls, that you can use for your comparison. Okay. Ion exchange chromatography falls into 2 different categories. There's cation exchange chromatography and anion exchange chromatography. Now, what this relates to is what is immobilized permanently on the column. So think about this as ion exchange chromatography is, well, it is a column, and there are small beads. And each one of these beads has a very small modification onto them. So let's draw this right here. And one of the questions I always like to ask is, let's say you're at a physiological pH, and you have permanently affixed a small molecule to it. What would be a good molecule that we've become familiar with in this class that you could permanently affix to it? Well, CH2, COO minus. That's the R group for aspartic acid. That is a negative charge. What is that going to be attracted to? That's going to attract positively charged molecules. So positively charged proteins, positively charged ions, anything positively charged will become immobilized on this. Now this is an example of a cation exchange column, or a cation exchange resin. So there's a small bead that has a modification to it. That small bead has this aspartic acid R group affixed to it. It will bind to positively charged proteins. The opposite is true for an anion exchange chromatography column. That's going to be a small bead plus CH2, and let's just go with CH2. If we had something like an arginine, so if we have an arginine R group, CH2, then NH, and then we've got our, C, double bonded NH2, positively charged, then an NH2 here, Well, now we've got this positively charged, that's not a positive charge there, I apologize. We've got this group that has an overall positive charge, which is going to attract negatively charged proteins. So this would be a good resin to have permanently present on an anion exchange column. Okay, so the names, you have to keep those straight. An anion exchange column will attract negatively charged proteins. A cation exchange column will attract positively charged proteins. What is affixed to those beads? That's going to vary from column to column. An affinity chromatography column is basically the exact same, except you would have something, you'd have these small beads and something affixed to them permanently. For instance, what you could do is you could affix glucose to it. So then what happens is, or any sort of sugar, any hexose or pentose, well, any protein or any molecule that has an affinity for this glucose or this sugar is going to basically grab onto those and stay there until you do something to move them. So affinity chromatography works based on interactions between protein or interactions between molecules. For our sakes, we say protein and a small molecule. So that's one of the key points of ion exchange chromatography, affinity chromatography, and gel filtration chromatography. What are you actually separating and why? Now, in lecture, what I went through was I talked about activity and specific activity, and I'm going to go ahead and revisit that on the next slide. So, a measure of purity of a protein is found in specific activity. And it's kind of intuitive when using the word specific, but you are looking at an individual protein there and the presence of that individual protein. So is your protein in the mix? Generally, whenever we look at activity versus specific activity, we are looking at activity that would be measured in units, generally the letter U. Specific activity, SA, is units per milligram. So how much of your protein is present in the total amount of protein there? So as specific activity goes up, so does purity. The purity of your protein goes up in direct correlation with the specific activity of a sample. So what you want to see is, or basically what you are doing is you are enriching for your protein, and that's an example, or that's how you will see your specific activity go up. So take heed of that, and thank you very much for watching. I hope this is helpful, and I will see you next time.